I'm going to touch on a few things, a few questions that we've got here. There's been quite a lot of comment on Neurone in the Facebook chat, so we'll be touching on that at some point shortly. And in the current climate with COVID-19, I think I'll just turn to Chris and ask him about his telemedicine. We have a question asking whether or not there's a space for wearable technology in clinical trials for data collection and tracking and reminders going forward. Thanks, Chris.
Thanks, Nick. So very much so. I think there's a place for remote technologies for both care and for research. So I think in terms of for care, being able to monitor an individual's respiratory function in between clinical visits, to be able to monitor how they're getting about at home, how monitoring their weight and having all this fed back to the centre so the centre can act if there's a new care need in between the clinic visit. So I gave examples of questionnaire-based things, but we are also building in peripherals. And similarly for research, so one of the statistics that often surprises is people that less than 10% of individuals living with ALS motor neuron disease go into clinical trials of the type that Jinsey's just been describing. And there are complex reasons around that, but one of them is the burden of taking part in a clinical trial and where people live, how easy it is to get to a centre that's running a clinical trial. If we were able to use peripherals, so home spirometry, and there are other gadgets for monitoring speech now, for monitoring dexterity. If we were able to use those in clinical trials, then more people would be able to take part. So I think the vision of many of us, if not all, is that everyone should be able to be offered the opportunity to take part in a clinical trial wherever they live, and that the burden of participating in a clinical trial is minimized.
Thank you very much. I guess that's one of the few positives that may have come out of the COVID situation, is this change in trial design and more accessibility. So if we can, we'll move on to a small comment about Neurone a bit in a moment, but I think you've all mentioned biomarkers, and I think we all realize this is a really important thing to really get a grip on. So I think, Ludo, if you could just shed some light on what you think your work on biomarkers and what you know of it and how that's helping your research, and then we'll just go through and see how that's being utilized in clinical trials.
Yeah, indeed. I completely agree. I think biomarkers are going to be very, very helpful, especially in selecting those therapies that really make a difference. And in that sense, I think it was already said during the presentation, it is very encouraging to see that in the tofersen trial, that there clearly also an effect is seen not only on the clinical appearance in the patients, but also on the neurofilaments as biomarkers. So I think that will also help, and I know that patients always have that question that they don't want to end up in the placebo arm of the trial. So I think if we have very, very good biomarkers, we could limit, I think, the number of people in the placebo arm, and that you can indeed increase the chance that you will see an effect in as many patients as possible. So I think it's difficult to underestimate the importance of good biomarkers. And having said that, I think we are going in the right way. I think we have some good biomarkers at present.
I think there's quite a large number of pieces of work on different biomarkers that are being presented at our symposium next week. I know neurofilament is really the front runner in this, and I guess all trials will hopefully be designed measuring neurofilaments going forward. So I think what we'll probably turn to now is the question of Neurone. So there's a great interest in this from the community, and it's a reflection, obviously, of the great strength of our community that there is such interest and an important need to find out exactly where we're going with all of the treatments and trials that are in preparation. So, Jinsey, do you want to just give us an overview of what we do know Neuron from Brainstorm, and I think that will give our audience some indication. Thank you.
Yeah, I think the big picture here is that we're still waiting for the details of the phase 3 clinical trial results from Neuron from Brainstorm. It was a clinical trial of 200 patients that were randomized either to placebo or to autologous mesenchymal stem cells. And it looked at a group of ALS patients in the US that were less than age 60, were less than 2 years in their disease onset and had a breathing of more than 65% of what's predicted for their lungs. And they had to have a minimum ALSFRS score of 25. So it was a subgroup that was evaluated based on the data from their phase 2 study. And just as a disclosure, I was not a clinical trial site in the US. So what's known publicly right now is what's been released in the top line press release, which interestingly, their primary endpoint was looking at responders who had a 1.25 point per month improvement on their functional rating scale slope compared to placebo. And their assumption was that 35% of patients treated with the neuron cells would have that improvement, and 15% of the placebo would have that improvement. But what we saw in the trial results was that about 35% of treated patients did show that response, but about 28% of patients that were treated on the placebo also had that post-treatment improvement in their slope, raising the possibility of a placebo response. So the primary endpoint was not statistically significant and considered negative. The secondary endpoint was more of a traditional evaluation on the ALS functional rating scale. So it was the change from baseline. It's the same data in my presentation. You looked at edaravone. I presented the change from baseline and also for the Amylyx drug, AMX0035. Now, when you look at all of the patients that were treated, the change difference in the treated patients with the neuron cells versus the placebo was a difference of 0.36. So less than what we have seen for edaravone or AMX0035. And again, it was not statistically significant. So that's all we know right now regarding the story. There is a mention of a pre-specified subgroup analysis, which showed some improvement of the slope in 35% of patients versus that were treated with neuron versus placebo, 15% of placebo patients. But we don't know how many patients were included, what the characteristics of that subgroup is. And there is, I'll just say that I'll put a plug in on December 9th at the International MND meeting. There will be a public presentation of details regarding the study. So I think we'll learn more about it and how to react to it once we know the details of what occurred in the study.
Thank you. So in terms of announcement, I think it means that the top line data has to be put out relatively quickly when it's available, and then the data will be mined in more detail to try and find and understand exactly what's gone on with that work. So yeah, we really look forward to that presentation at the International Symposium. And it's really a reflection of the kind of positivity that there are lots of trials that are reading out at the moment, and we're feeling that we're going to get answers to a lot of these things. Janice, you spoke about the SOD1 gene and the tofersen trial. So that will be useful extremely useful if it works and is positive in a small subset of people. So are there other bits of work going on with sporadic patients in that same gene technology arena?
I think probably Jinsey and Chris would be able to speak to this better than I can. But the question is whether or not SOD1 is involved in sporadic ALS. Some people feel that it might be. Whether or not there's going to be a clinical trial of tofersen in sporadic ALS, I'm not aware of that at the moment. I don't know if Jinsey and Chris have any more thoughts on that.
Sure. Do you want to take that, Chris?
So I don't know if there is going to be a trial. I know there has been talk of it in sporadic ALS. I think there is a school of thought that SOD1 and misfolded SOD1 is part of certainly a subgroup of sporadic ALS. So there is a sort of a rationale for doing it. So I wouldn't be surprised if a trial went ahead.
Yeah.
And I think what we have to do though is confirm that it works in actual SOD1 ALS first of all, and if we see that manipulating, knocking down mutant SOD1 is beneficial, that adds more sort of interest into, okay, well then what would it do in sporadic disease? So I think there's some learning to be done first of all.
Yeah, thank you. And there is other work going on with other targets like ataxin-2 as well, which should hopefully give benefit to many people on that. That's great. So a question again, probably Jinsey and Chris, about trial design. So really it's asking the question whether the rules are different between the FDA and the EMA, so the European Medicines Agency, and the FDA in the US, and whether trial design actually takes into account those regulations and how approval is met by them. Do you want to take that, Jinsey, please?
Yeah, I think I'll start just from the perspective of the US FDA review. I think from their perspective, regulatory perspective, they did provide advice that we were able to use the ALS functional rating scale as an endpoint. We would have to look at survival, but I think we were given a little bit a shorter duration of observation for the controlled portion than typically would be required for regulatory authorities outside of the United States. So we have our platform trial has a 6-month duration to answer the question of whether any potential therapy is effective or not in that disease population. So I think that might be one key difference between the US-based platform trial and maybe other platform trials. I don't know, Chris, if you have any comments.
Yes, so that's exactly it. From a regulatory perspective, if it's a regulatory study for approval, so in old money, phase 3, the safety data needs to be over a longer period of time in particular. So the reason that edaravone hasn't been approved in Europe, I think, relates to the duration of the trial and confidence in the safety, as well as short duration of the outcome measure. ALSFRS is allowed as an outcome measure, primary outcome measure in Europe, but there does also need to be an additional functional measure and survival as secondary if ALSFRS is first. But I think it's the duration that's the key difference, and why we have this issue over edaravone in different territories. It would be good if there could be some harmony across the regulatory authorities, and I suppose that's up to patient groups, advocacy groups, researchers, clinicians, industry to come together and lobby for some form of harmonization, which I think would be good.
Thank you. So one back to COVID-19. I think, Chris, you may know something about this. I wasn't aware of this, but early mouse studies are showing that coronaviruses, including COVID-19, cause long-term damage to neurons. Do you believe the pandemic may cause a spike in ALS and other neurodegenerative diseases in 5, 10 or 20 years time in the future? I guess in answering that, maybe you could just touch on a few of the issues with COVID-19 as well. Thank you.
Okay, so I'm not aware of the mouse data myself with regards to COVID, so if anybody else is, they can shout up afterwards. I think we're learning so much about COVID and its effects on individuals, both acutely, but then the long-term effects and the so-called long COVID. We know that it does have a predilection for the nervous system. So anosmia, so the loss of smell and altered taste, shows that it's going straight in there and affecting nerves, and getting into your CNS through the olfactory passages is a good way in. So I wouldn't be surprised, but it's just conjecture at the moment. In terms of impact on people with MND ALS now, I think I touched on the way care delivery has changed, and trial delivery is changed, in the last year. We have, I think, patients so in the UK, are self-isolated and shielded is the phrase that's used, and been perhaps reluctant to go to the clinical centres, so been reliant on those more remote technologies. It's variable. We've done surveys to find out how has diagnosis been delayed, has gastrostomy insertion been delayed, as respiratory assessments, and it's mixed really. You see a mixed picture. There are some areas where I think care has been impacted with diagnostic delay and delay to interventions, but other areas have managed to adapt their services and been able to carry on as some form of normal.
Thank you very much indeed. Ludo, you talked about modelling the disease, and kind of one of your goals will be to have this all different cells, three-dimensional model of ALS in a dish. How realistic and how far are we away from that goal?
That's a good question. Yeah, the reason, maybe I should first explain that, the reason why we also want to incorporate other cell types in our model is that it's becoming more and more clear, and especially the work of Don Cleveland that I should mention here, that motor neurons don't die in isolation, but that also non-neuronal cells can have a huge impact on the disease process. And so in order to find out how these non-motor neuron cells have an impact, I think we need to add them to the system that I just described. So we have protocols, so it's going quite fast. So we have protocols to differentiate all these different cell types starting from iPSCs. We can add them one by one. The major problem is more a technical one. Every cell type has special requirements regarding the medium. So if you start to combine them, you have to make a compromise. So for the moment, we are adding microglia and astrocytes to our system, to our neuromuscular junctions. And by the way, that already gives us very interesting insights. Adding even more cells. I'm not saying it's impossible, but then we need to find a medium that fits them all. So we have to give them the factors that can keep them in the differentiated state that we want them in. But I think it's not impossible with all the new technical achievements that we have related to the stem cell research, actually, which I think has helped us a lot. So I hope it's going fast, but it's always taking a bit of time.
Yeah, that was beautiful imaging and absolutely incredible that you can make motor neurons from patient skin cells. It's science fiction, and it's a credit to the dedication and the generosity of patients and people affected by MND that provide those samples. Thank you.
Yeah, absolutely.
Janice, you did a wonderful job of explaining the SOD1 aggregation and that mechanism. Could you explain possibly if there's similar mechanisms that cause ALS MND from other genes in a similar way, or is it far more complicated than that?
Well, the SOD1 mutations cause the SOD1 to misfold, and that's very clear for the ALS cases caused by mutations in SOD1. And as we've alluded to already, that there may be misfolded SOD1 also in sporadic ALS. One of the key things that the studies of the SOD1 has given us, as Jinsey had mentioned, was we've learned a lot about the biology of the disease from studying SOD1. What are the downstream pathways that SOD1 activates that we can target therapeutically? But in addition to that, there are other types of misfolded proteins in sporadic ALS, the most common of which is a protein called TDP43. And that is present in 97% of all ALS cases. And so a number of groups, including our own, are looking at therapies to target misfolded TDP43 as a therapy for sporadic ALS and for other genetic forms of ALS, including C9orf72.
Thank you very much indeed. Ludo, again, you mentioned humanized zebrafish. Do you want to just explain what that means?
Yeah, well, it's still early days. I think what in some situations, or what we want to do next, of course, is to see how our iPSC-derived motor neurons behave in an animal. This animal could be a mouse or a rat, but that is, not so easy. The big advantage of the zebrafish is that they are translucent. So you can inject a human motor neuron in a relatively early stage of their development when their immune system is not yet fully developed. And then you can see how that human motor neuron survives or integrates into the zebrafish. And then, of course, you hope to see that an ALS motor neuron is doing that in a different way than a control motor neuron. So the big advantage of the zebrafish is that they are translucent, so that you can follow it live. So you even don't have to kill the animal to see what happened with your human motor neuron. So that makes it interesting to see how it's going over time. In addition to the fact that the immune system is helping us there as well, as it is not rejecting non-zebrafish material at a young stage. So it's too early to say that it works, but I have good hopes, and I have some preliminary data that give me some courage to at least mention it already in a presentation. So
thank you.
More to come.
Great. Chris, you mentioned people entering the right kind of trial for them. I'd like to just touch on that again in a moment. And Jinsey, there's a question following on from that really, which is how should a patient decide whether to continue in the open label phase of a trial or move on to another compound? Chris, if you want to take that right type of trial comment first, please.
Yes, so part of that is aspirational. So there's an idea whether MND is all one disease, all the same disease, or whether it's actually lots of different diseases that look the same at the end. The result of the motor nerve damage is the same. There are different ways of getting there. Now, we can't do what we want to be able to. So I do believe it's different diseases. And I think the fact that we're just talking about these genetic diseases and different genes is a very good example of that. And then on a pathological basis, SOD1 is actually very different to the rest of, well, almost all of the rest of motor neuron disease. And the rest of motor neuron disease is a TDP43 protein disease, whereas SOD1 isn't. So there's a difference. So SOD1 patients need to go into trials that are targeted towards SOD1 and things that go wrong in SOD1 type disease, and that's what's happening with tofersen. So tofersen is a brilliant example of where we need to be going. We've got C9orf trials now for people with C9orf MND disease, so that's happening. So we're getting there with those. The sort of 10% of people who have genetic disease. What we need to do now is find a way of separating the sporadic disease, because I believe, as many others do, that within that sporadic disease, you've probably got people where the axonal transport machinery is the thing perhaps that's predominantly affected. So those lovely cartoons that Ludo showed. You might have others where it's the energy generation in the cell that's wrong, and that's the predominant problem. And so the sort of fantasy, which I don't think is too far off, is that we begin to be able to dissect that. So, for instance, the hyperexcitability, we're beginning to see that there are a subgroup of patients who have this hyperexcitability in their cortex, and a treatment such as the retigabine, might be more beneficial in those. You don't want to give it to patients who don't have the hyperexcitability. So we need to be smarter about how we subgroup patients, find out what type of MND they have going on at a biological basis. And we aren't there yet, particularly for the sporadic disease, but we're making progress, I think. And I think that's the next big breakthrough.
Thank you. And Jinsey, do you want to take that one on the open label choice?
I think that's a pretty common question I get asked. And over the last 15 years, I've had patients kind of come up with their own algorithms. I can't even take credit for it. But, when the landscape gets crowded with a lot of clinical trials and a lot of options, I think that's great for people living with ALS. But there are a few things to consider. One is when you complete a clinical trial, there may or may not have been a placebo and a chance of that. And that chance can vary from clinical trial to clinical trial. And when you get the informed consent for a clinical trial. When the doctor asks you, are you interested, and provides you a sheet that's called the informed consent in there, and it should be discussed and you should ask what the chance of getting placebo is. And so that can be 50-50, 25%, 33%, it could be a variety of things, or zero. And usually after that placebo-controlled portion, there will be an open label extension. Open label extension is usually provided to clinical trial participants first. So you have an increased access to that investigational therapy where you're guaranteed, and you will know you're getting that investigational drug, your physician will know, and the sponsor will know. So it's essentially reciprocation for your generosity of participating in that particular clinical trial, to answer the question. Now, usually you are provided that option before we know the full results of that clinical trial. So it is difficult to make that decision. So you might come to a point where you're offered open label extension, where you'll know you'll get access to drug and there's zero chance of placebo, or you'll get an opportunity to participate in another clinical trial. And so one of the most important questions people ask is whether there's going to be a placebo if you move to another clinical trial. And so it's a personal choice for people living with ALS, whether they want to go into another clinical trial or stay in an open label extension where they're guaranteed to get that investigational product. And some people may choose the clinical trial because it might have a really great data, preliminary data, showing a better effect than what was anticipated for whatever they're going to get in open label. But other factors that play a part is the timing, right? So timing of when that next clinical trial may come. It may not be there, so open label extension would be the better choice. The route of administration, is it easy to take? Is it oral? Is it IV? Is it intrathecal, where they have to do a procedure in hospital? And with that, how often would you have to travel in hospital, or can you remain at home? How many assessments? Usually in open label extension, there's far fewer assessments and far fewer things that the patient would have to do or the caregiver would have to do. And then they would just have to figure out whether that would be the right fit for them, because some people even choose to decline participating in the open label extension because during the clinical trial controlled portion, they might have had an adverse event or they feel like they didn't have any effect. So that's kind of a big picture overview of what you might consider when you're making. But data is never there when these decisions have to be made. You have to look at what's there to date and talk about what risks you want to take. And risks are not just the adverse effects for the drug, it's the time commitment to participate, it's the burden on the caregiver and travel and finances. So I think those are the key factors.
Thank you very much. We're coming quite close to the end of this time. So what I'd like to do is just go across the panel shortly and ask you for your kind of message of hope for going forward, because 2020 has been a pretty poor year so far in many ways, but there are some optimistic things on the horizon. But just returning to you, Chris, while we've got a couple of minutes talking about diet, I think that's really something that everybody is something that people can do at home, I suppose. So do you have any kind of take homes of your learnings from your studies, in particular also things like the microbiome has been mentioned in the media, etc., in association with ALS? If you'd like to just touch on that for us, that'd be great. Thanks.
So two questions there. So the first one, just to do with diet in general. I think the only thing that we have a sense of is that losing weight is bad. So try not to lose weight. So, I was to say one thing about what people eat is eat enough so you don't lose weight, and if possible put it on and ignore healthy eating messages. Fatty diets, things like that, no evidence for, don't go there. Just have lots of the good stuff. That would be the message, and seek help from dietitians and speech language therapists, to make that possible. In terms of the microbiome, it's an evolving area. It's not without its controversies, it's fair to say. In terms of it's sort of humans, because there's a lot of work and controversy with the microbiome in the mouse and the effect it's having on mouse models and how we interpret results. But in humans, we often talk about all this variation between patients. Why does some people live? 5% of people alive for 10 years. Why do some people have terribly aggressive disease? within 6 months passing away. So there are other variations within the disease, and we look to our own sort of genetics and genes to try and understand that. But there's another huge amount of genetic information, and that's the microbiome. So that's all the microbes that we live with or live with us within our GI tract. And they're secreting different chemicals, compounds, enzymes that are being absorbed into our bodies and having, we think, a much bigger influence on our metabolism and how our cells function than we ever realized. And what we're trying to do now, and there are several groups looking at this, is seeing, is this the environmental factor that might explain some of the in-patient variation? Could it be a factor that influences how drugs are absorbed, different microbiomes? And there have been some studies where people have tried to alter microbiomes within mouse models, and have shown when they alter microbiomes to a particularly favorable microbiome, they can get a better outcome in that model. I would say it's very early days, and there's a lot of work to be done, but it's that we now have the technology to look at this in detail now and begin to unpick it and understand, is this one of the big environmental factors that we need to understand.
Thank you very much. Okay, so I'll just pass along line. There's a question actually about an update on the cooperative SEM trial. Does anybody know of that? I believe that it's almost finished recruiting and is going to read out end of Q4 2021. Yeah. Okay. So let's just leave with your optimism message of hope for the next few years or so. Chris, are we heading in the right direction, do you think?
Yes, very, very much. I think so. I think we're on the verge of a treatment for at least one type of motor neuron disease, I hope. So there's a lot of optimism around gene therapy for SOD1 disease. And if I just think back 10 years ago, I didn't think that we would be at this point where we are now. I think we've got there because of all the research that's gone on leading up to this point. And if you widen that lens, the research that's gone into sporadic disease, we now have a much better understanding of what's actually going on within the cell, more than we've ever done. And so I think we're going to build on that quite quickly now. We have the infrastructure, the trial infrastructure to be able to test compounds quickly. We're going to have better, we're going to have biomarkers so we can do shorter trials. I think there's going to be a rapid pace of change. It can't come quick enough, but I'm hopeful that it won't be another 10, 15 years before the next big breakthroughs are being made.
Thank you very much. I think we're into a yes/no answer here. Optimistic, Janice?
I'm incredibly optimistic. I think that the tofersen trial has shown us that genetic causes can definitely be treated. And in fact, for all of the neurodegenerative diseases, in my understanding, this treatment is the first for any of them. I think Huntington's disease is another where they're doing the gene targeting. The thing I'm incredibly excited about is the microbiome. The fact that you can take, it's been long known, the mouse models, you move them from one part of the country to another. Some mice live longer than the others, and it's all due to diets and the bacteria that's in your gut. And they're able to assess the bacteria in your gut. And I think we're going to hear a lot more about that in terms of ALS and other neurodegenerative diseases. So I'm incredibly excited about that because that affects everybody.
Great. Thank you very much. Ludo, Jinsey, yes, optimistic. And we're going to have to stop now. I think we're at the end of our time. We could have answered many, many more questions and spoke for a considerable amount of time. And just want to thank everybody for joining us here live, and for those who've managed to catch up with the recordings. It's been very interesting to see that work, and I think it's important to stress that we're all on the same page. Everybody's trying to work really, really hard to understand this disease and find treatments and effective therapies, and we can't do it without the patients and their caregivers, and it's a really united effort. So I think, we're going to take that at heart and all move forward together. Thank you very much, and I'll hand back to Tammy.
Thank you, Nick. We're so appreciative of all of your efforts to coordinate the panelists today and for their very important messages. We know how much ALS M&D Connect is appreciated by both our Alliance members as well as the people within our community. It's an opportunity to get to ask those really specific questions. And I think this morning's panel was a great example of showing how we've moved through time to be able to get to the discoveries which are now leading us to these very promising therapies and what they mean and how much more that we know. And of course, it can never come soon enough for our community. Yesterday is still too late, but people are working so hard to be able to get us to that very important pivotal point where we've got therapies that are slowing progression and getting us to better understanding so that we can make the moves we all want to see on ALS for a future without ALS.